Okay guys, I'm going to start this new unit with transport, okay? So when I say transport, I'm talking about how substances get into and out of cells, okay? So that's what we'll just generally, biologists will refer to that as transport. So there are several different ways that that can happen. I'm going to take you through each of these. You need pages 1, 2, 3, 1, 2, and 3 from your booklet. Okay, I know the booklet, the quality is not fabulous, but I will, when I have the notes handy on my USB stick, I'll upload them again onto the LMS page. And if I don't have them because they're on my school computer, then we'll be looking at these pictures. Okay, so a lot of this will be review and some of it hopefully will be new to you. So one type of transport is diffusion. Okay, so I just want to show you something. I'm going to move up to my computer here and show you the screen. You have this picture, sorry. Okay, and so this is something that you currently have also on your LMS page. So cell transport can be divided into passive and active transport. So I'm going to start the lesson out with passive transport, and we'll highlight the fact that it doesn't require energy. But as we're going through this note, I keep this in the back of my mind, okay? There's passive transport. It can be divided into simple diffusion and facilitated diffusion. So I'm going to take you through those also. And then simple diffusion involves osmosis. I'll talk to you about these terms a little bit, but I wanted you to see them on here. Use this table also to add extra definitions of these terms. That might help you out. We'll go through facilitated diffusion where we have got channel proteins. I won't talk to you too much about carrier proteins because this is a grade 12 topic, okay? And then when we go to the opposite side, so passive transport doesn't require energy. So you might want to write that on this line, does not require energy. And then when we get to active transport, this requires energy. And there are two types of active transport. There's active transport that involves vesicles and some that involves pumps. I won't be dealing with pumps because this is also grade 12 work. Okay, so in grade 11, we warm you up with vesicles, exocytosis, endocytosis, etc. Okay, so if I don't touch on pumps, that's because it's something I talk to the grade 12s about. And if I don't touch on carrier proteins, that is again something I talk to you more about in grade 12. I'll give you specific examples. Okay, so back to the note. All right, so movement across cell membranes. All right, so we've got diffusion. Diffusion is passive. When you see passive, that means it doesn't require energy, okay? It's going to happen on its own. So in terms of passives, I just have to get to a point, sorry guys, where I can see my hand. Diffusion is the net passive movement of particles. And passive is when we go with a concentration that's higher to a concentration that is lower. So particles are going to move from an area of really high concentration. So if you've got lots and lots of particles, okay, jam crammed in a small space with a time lapse, eventually we'd say maybe 30 seconds later or 30 minutes later, the particles would be spread out, okay? So they go from high concentration to low concentration, all right? That continues until the concentration is uniform. In other words, we would have equal spacing between these particles. Something you learned about in elementary school, all right? This requires no energy. So passive transport requires no energy. You don't have to give this system anything. The particles don't want to be cramped. If they give enough room, they will spread out from each other, okay? So remember that this is passive, not just this picture, but what we're talking about right now, this is passive transport. And so one type of passive transport is what we call simple diffusion, okay? So in terms of your flow chart, this would be simple diffusion. All right, so where are we going to see this? Well, why am I starting with this? Because we need to talk about this when we get to the respiratory system. So it's going to happen in the alveoli in the respiratory system, okay? And I'm going to make a big deal about that when we get there. It's going to happen in photosynthesis. Okay, so when we get to plants, we're going to talk about carbon dioxide from the air diffusing into the leaves. Why? Because there's a higher concentration of carbon dioxide in the air than in the leaves. There's a higher concentration of oxygen in the air than in our blood. So that's how diffusion works for us. Okay, so we will highlight that. And then gas exchange and respiration. This is going to be important because We touched on it in grade 10, but in grade 11, I'm going to show you where this happens, and it's going to be something you need to know. So oxygen from the blood goes to the tissue cells, and carbon dioxide in the tissue cells goes to the blood, okay? Those are going to be some main topics, and that's why I start with this today. Don't worry about this, but I want you to know that osmosis is another form of diffusion, okay? It's diffusion of water, and because the movement of water happens so frequently, we give it its own name, all right? So osmosis is just simply the diffusion of water, all right, through a membrane that's semi-permeable, and it allows water to go through it. All right, how do we increase diffusion? So that's what we could say this title is, how, whoops, sorry, I'm out of focus there. When my hand gets out of the way, you'll be able to focus again. How do we increase diffusion? Okay, that's what this answers here. So if we have a short distance, diffusion is going to increase. If we have a large surface area, diffusion is going to increase. If there's a big concentration difference between particles, that's called Fick's law, diffusion is going to increase. If we increase temperature, particles move more quickly, diffusion is going to increase, okay? How do we decrease diffusion? Larger particles move more slowly, all right? So increasing surface area means that we've got a larger area than not, okay? And so that's going to be coming into effect when we talk about the lungs, okay, and the alveoli. All right, so that's one type of diffusion, just simply particles spreading out because they're too crowded and they can move because we're giving them more area to move, and they're moving from an area of high concentration to low concentration. That's simple diffusion. The second type of diffusion is facilitated diffusion, okay? So in facilitated diffusion, we have particles moving from an area of high concentration to low concentration still, okay? But this time they're going to pass through a carrier protein. So we have movement of particles from high concentration to low concentration using a carrier protein. So when you see the term concentration gradient, it means we're going from a high, square brackets are symbols for concentration, to low concentration. That's what we refer to as with the concentration gradient. So diffusion always goes with the concentration gradient or down the concentration gradient is another way of saying it. We're moving from high concentration to low concentration of particles. Now, sometimes these particles are just too big to get through the phospholipid bilayer, okay? So here is your phospholipid bilayer. All right, we have a phosphate head and two fatty acid tails. So phosphate head, two fatty acid tails. And when we get a whole bunch of these guys together, we call that the phospholipid bilayer, okay? Normally I would introduce organelles once again, but at this point, with the pandemic, we're not going to spend any time on organelles, okay? So just understand that your membrane is made up of a phosphate head, two fatty acid chains, and then spread throughout that, you've got these proteins, okay? And one type of protein that we're looking at today, a particular type of protein, is called a channel protein, all right? And the function of these channel proteins is this. They function in facilitated diffusion. So notice we've got a high concentration of glucose. Okay, I'm going to color glucose in green. All right, high concentration of glucose on one side of the membrane. So you could pretend this is the exterior of the cell. Okay, and so this cell would like to take glucose in. This is the inside of the cell. That's the outside of the cell. So what does that mean? In terms of, I know that sometimes people have a hard time visualizing this. This is the cell you would have drawn when you were in elementary school, okay? You would have labeled that cell membrane. Now, this is your cell membrane. This whole thing is your cell membrane. So what we're showing you up here is a little section of this. So this would continue and go all the way around the cell, okay? So that's what you're technically looking at here. So you have lots of glucose outside the cell and not very much glucose inside the cell. And that's the situation we're looking at. So glucose is too big, and for other reasons as well, but it's too big to fit through these phospholipids, okay? They're not going to let it get through there. Oxygen can go through there, no problem. Water can go through, because when you're talking about oxygen, you're talking about two little molecules stuck together, okay? these are 6 molecules right there, plus all the H and the OH groups coming off. This is a very large molecule compared to oxygen. It has to go in through a channel. And so that's the purpose of these proteins. They serve as channels. So glucose is in a high concentration out here, low concentration inside. And how does it get through? It gets through this channel. So notice the shape of the channel is specific to glucose. So every large molecule will have a different type of protein channel that will allow it to go through the membrane, okay? And that's what I want you to understand in grade 11. All right, so each carrier, each channel has its own shape. Now, they're calling carriers something different here. A carrier protein, just so I show you, I said we're going to study this in grade 12, but a carrier protein would be sitting on the outside of this membrane, okay? And the purpose of the carrier protein is it would pick glucose up and then it would carry it over to the channel protein, okay? So that's what a carrier protein does. It picks up a specific molecule. So for fats, you would have a different carrier protein than you would for glucose. You would have a different carrier protein for sucrose than you would for glucose, okay? And by the same token, you'd have a different channel protein for glucose than you would for fructose and sucrose. All right, everything's very specific. So what you need to know is facilitated diffusion means that you've got helper facilitating proteins that are helping to change the concentration. So in other words, glucose is in a high concentration here, it's in a low concentration here. It wants to spread out, it wants to go down the concentration gradient. How does it do that? It does that using a channel protein, all right, because it's too large to get through, all right? Important, it's passive and it requires no energy, okay? Don't worry about this. That's something I would go over in grade 12 with you. So as long as you understand the concept here, you can read through this. It's all very good, but I've outlined the concept with the diagram. Okay, so next page, as we flip this over, we go through osmosis. So specific to diffusion, and this is simple diffusion. So going back to number 1, simple diffusion. Okay, so and that what I'm referring to that table I gave you, that diagram that I've given you, okay? So this is simple diffusion, and simple diffusion includes osmosis. And what is osmosis? It's the diffusion of water, okay? So diffusion and osmosis are one and the same thing. They require no ATP. What is ATP? It's adenosine triphosphate, and it's the energy source that we use for everything in the cell, okay? So if we had adenosine, and if I would have reviewed with you your organelles, I would have made you learn this in the organelle section for mitochondria, because cellular respiration makes ATP. So what is ATP? It's this adenosine molecule. You're going to learn what this is in grade 12, biochemistry, okay, so biology, and it has 3 phosphate groups attached to it. Okay, so it's got 3 phosphate groups, one after another. I was making it long, it didn't have to be that long. 3 phosphate groups. So in other words, you've got adenosine, and there's a phosphate, and a phosphate, and a phosphate, and this is what we call ATP. Okay, if you sever this bond, that's ATP. If you sever it, and when I cut it, then it goes to ADP, adenosine diphosphate. Every time you cut a phosphate to phosphate bond, you release huge amounts of energy, and that powers your cells, okay? So why did I tell you this? Just so that you understand, ATP is the source of energy in your cell. You take all your food and you convert it into ATP in the mitochondria. So the mitochondria does that for us. Those little organelles that came to us via endosymbiosis. All right, so simple diffusion, osmosis is just a form of simple diffusion, and we're talking about not using any ATP, all right, and it's the movement of water, okay? So in other words, if we have, just this diagram is pretty good, so I'm going to show you. If we have salt here, and salt is purple, and we have more salt in the cell that's beside that cell, okay? So we've got a cell that's taken in more salt, maybe you had some salty meals, and then this cell has taken the salt in first. Osmosis will do this. We don't move the salt as much, and we don't have a way of actively transporting water. So what happens is osmosis takes water, and we can put water in blue, H2O, and we can say we've got some water in here, right? Because you've got water in your cells, and we've got water in this cell. okay? And because this cell has so much salt, what's going to happen is the water is going to move from that cell to this cell via osmosis, because there's a higher concentration of sodium in here, of salt, than there is water, and water will move through osmosis to decrease that concentration of salt, okay? So that's something you need to understand. When we have a concentration difference of certain particles like salt between one cell and another, water will move, and that's where osmosis comes into play. In order to decrease the concentration of salt here, what it does is it moves in and dissolves the salt, okay? And so we increase water here, and water would move in that direction. All right, so some major examples of osmosis: absorption of water in plant roots. I'll get to that when we get to plants. That'll be something I will emphasize. Reabsorption of water in your tubules of your nephrons. So I'm not going to highlight that. So this occurs in your kidneys. If you have a very salty diet, you're going to take in a lot more water. You're going to reabsorb water. Okay. Reabsorption of tissue fluid into veins of blood capillaries, at the ends of blood capillaries. So again, sometimes you can save your water in your blood. Okay. So if we've got some concentrations that are too high, then we will do that. We will reabsorb water, okay? And it's based on this idea here. An absorption of water by the alimentary canal. So in other words, if you're not drinking enough water, your colon will absorb more water from your food, and then you will have dry feces in order to conserve your water. So water will move from the feces back into your body because your body doesn't have enough water. All right, osmoregulation is maintaining your water balance. Okay, so osmoregulation means to keep the concentration of your water at a rate or at a certain concentration that's important for your body to be able to function. Okay, in terms of humans, we do this in our kidneys. All right, so I'm not going to spend too much time on that. What I'm going to move over to now is osmosis in plant cells. So just a few terms that you need to know, especially to do your homework. We're still on osmosis. This is a plant cell, okay? And you can read through this here. I'm going to teach you with the diagram. So we've got our plant cell, and we've got the vacuole. Now remember that the vacuole contains water, okay? So the vacuole contains water, all right? And a plant ideally would love this. So this is the cell wall, okay? And then you've got your membrane, and this is your cytoplasm. Plants love when the vacuole is full and the water pressure is pushing against the cell wall, okay? So plants like to be in a state like that, and that's said to be turgid, okay? So the pressure from that filled vacuole pushing against the cell wall is called turgor pressure, okay? And that makes the plant very happy, because what it does is it makes the plant stand up straight. The water's pushing against the cell walls, and it allows the cell to stand up straight. Now, I'm going to ask you a question. If a plant doesn't get enough water, what does it do? Okay, and your answer in your head or out loud might be you said it wilts. And that's exactly right. So when a plant wilts, what happens is, here's your vacuole, okay? And there's not so much water, so we're going to fill this up with lots and lots of water. Okay, and here there's not too much water. Notice the vacuole is not full, and there's not enough turgor pressure. Okay, so what happens here is the membrane is pulling away from the cell wall, and now there's not any pressure against this cell wall, so it wilts, it falls over. Okay, and when that happens, the plant is said to be flaccid. And that's not a good thing for a plant, okay? So for a plant, if we're talking about plants, the plant is very unhappy in that situation, okay? And so plants like to be in this situation where they are turgid. They don't like to be in this situation where they are flaccid. And that's what I want you to understand, okay? When the cell does this, when the cell membrane, the plasma membrane pulls away from this cell wall, that's called plasmolyzed. All right, and that's in bold for you. All right, and so turgor, just to finish up here, turgor is the pressure of a swollen cell against the cell wall, okay? And that is a really good thing. It helps the plant to stand up, all right? So it offers support, okay? When we get to plants, it changes the shape, and if you're wondering if we're going to get to Unit 5, we are. I will be able to grab about 2 weeks in Unit 5, okay? for you. So I'm not concerned about that at all. Considering exams have been eliminated, we're in good shape. So we're changing a shape of guard cells. We will get to that, okay? And I won't worry about plant cells maturing. Water potential, we're not going to worry about any of that. We're moving into active transport. So now we're moving into the other side of that table that you have in front of you. Okay, the flow chart, I called it. So active transport requires energy. We are moving against a concentration gradient. So in this case, we're moving from low concentration to high concentration. And you might ask yourself, why in the world would we do that? Okay, why don't things just spread out? Well, one specific example is in plants. So if you've got your plant, and again, we will review this when we get to the plant unit, but if you've got your plant and you've got your roots, okay, and so this here is the ground, and we're in the soil, and plants would love to take in water, okay? They like to take in water because of that trigger pressure I just mentioned to you. But no living thing that we know of in 2020, this is an important point, has figured out a way to actively transport water. So in other words, to move it against the concentration gradient. So plants and animals alike, okay, and all living things, what we have to do is we have to actively transport something else. In the case I showed you salt earlier. In this case, we're going to say the plant is transporting minerals, okay? And why is it transporting minerals like iron and zinc, magnesium? Because if it increases the concentration in its root of minerals, water naturally follows to dilute. So I made that point earlier here for that reason, knowing I was going to get to this topic, okay? So water will naturally dilute something, it'll move in, and that's the only way we know of to get water into something. We cannot actively transport water. We actively transport something else, and water will follow to dilute that something else. So that's an example of how plants do that. And so the water goes in to dilute the minerals, and then the water is able to move up and through the xylem and phloem, okay? So that's active transport and why something would want to go through active transport. So in terms of humans, we could actively transport glucose because we want to take it from an area of low concentration to high concentration, or sorry, high to low. No, sorry, I'm mixing that up. We want to move it from an area of low concentration to high concentration because we always want more glucose. Typically, our brain uses it, our body uses it all the time. Okay, so that's an example. Amino acids as well. Depending on need, sometimes we have to bring them into a cell that has a high concentration, higher than the outside of the cell, but that cell still needs more of it. All right, sodium potassium pump, I'm going to teach you about in grade 12, so we're not worrying about that. So this is the movement. When we get to endo and exocytosis now at this point, these are two different types of active transport. Okay, so if you recall on that diagram, I showed that to you. Two different types of active transport, and what you need to know is that these guys use vesicles and try and bring certain substances in. So in other words, the cell will open up a vesicle, and this is where I'm going to end teaching you guys with this. If we look at a cell, we could say this is an amoeba, for instance, or it could be an animal cell, but typically this is going to be an amoeba. An amoeba wants to eat, okay? So it finds these food particles outside of itself. Now what it does is it opens up the cell membrane so that it forms this pocket, and eventually this pocket will become a vesicle. So we're seeing the food here, stage 1. This is the first caption, time lapse. This is number 2. And this thing will form all around it. So the membrane will extend. And in time lapse number 3, we're seeing this a little while later, we've actually formed that into a vesicle. Okay? And that's called endocytosis. That's how cells eat. They can get rid of waste products just the opposite way. So if this was a waste product and we want to get rid of this, then the cell would bring this over to the edge, open that up, and expel it. Okay? So these guys are ways of transporting particles. And it requires energy, 100% requires energy. So this is the movement. When we talk about endocytosis and exocytosis, it's the movement of particles that are very large. Endo means the particles are going to come in. And exo means the particles are going to leave. So again, we're going to, this will be out. So this is getting rid of waste products. Carbon dioxide is a waste product also, but that will diffuse out. So waste products like animal waste, okay? Liquid waste, solid waste, and then particles that are going in would be food particles typically, okay? And so a couple different diagrams showing you that here. You can read through this here. It says it involves the fusion of the vesicles with the cell membrane. I showed you that here. Bacteria entering macrophages, substances destined for secretion are packaged in the Golgi apparatus first. So the Golgi apparatus would package this first and then bring it over for exocytosis, okay? A macrophage is a white blood cell that would eat bacteria, okay? So just so you know, you've got white blood cells that are quite large. We're going to go into that when we go into the circulatory system. And what they do is they go through endocytosis when they encounter different pathogens in our body. So they will physically do this, and then they bring this bacteria into us and they digest it, okay? So that's how your white blood cells work very frequently. All right, and lastly, the division between active transport, vesicles, exocytosis, and endocytosis. So when we're distinguishing different types of endocytosis, which is bringing something into your cell, you can either call that, if it's a liquid, pinocytosis or phagocytosis. So we often say pinocytosis is cell drinking. In other words, this would be a liquid coming in, and they're kind of showing you that over here as in droplets of liquids. And then when it's a whole particle or many particles and they're solids, we call it phagocytosis, and that would be cell eating.